Okay, good afternoon, everybody, and you're all very, very welcome to this talk. So John O'Sullivan is my name. I'm the current director of the Centre for Water Resources Research in UCD, the Dooge Centre for Water Resources Research. And this is a collaborative talk then between the CWRR and the Earth Institute. So I'm delighted to introduce Professor Martin Doyle, who'll give the talk. Martin has a background in fluid mechanics, sediment transport, a broad background that also incorporates infrastructure finance and indeed the development of water policy. And of course, that reflects his affiliations. So he's currently affiliated to Duke University in the US, but has spent time with the US government contributing to policy formulation in that respect. So welcome everybody in this room, and also welcome to those that are joining us online. And I'll hand over now to Professor Doyle.
All right, thank you. Mike, okay, do we know? I assume. Yeah. All right. All right, so thanks for joining. This has been great. I haven't gotten to think and talk about sediment in quite a while. So for the 7 of us that are interested in sediment, this is your day. But what I want to talk about today is something that kind of sprang up from 2 events. And it's mainly thinking about how we think about things. And so the first was a catalyst from my academic grandfather, and so my advisor's advisor. And one of the pieces of advice that he gave me was, when you're doing work on rivers or work anywhere, whatever you start with as your assumed reach, shift it 50%, and that's probably the best way to come up with a new idea. So at the time, everybody was working from, said, well, we need to study sediment mechanics, and we'll go from the cleanest reaches from the tributary junction to the next tributary junction. And he said, well, just shift it 50%. And if you shift it 50%, all of a sudden the tributary junction becomes the center place of your research. And this whole new branch of kind of fluid mechanics kind of came out of this thing. So he just said, always look for shifting your research 50% off what everybody else is doing and see what comes out of that. So I had this kind of floating around in my mind. And then I read an article in the newspaper about this container ship that had spilled, and within one of the containers was a bunch of rubber duckies, and it was in the middle of the Atlantic Ocean, and a bunch of oceanographers started to find rubber duckies scattered around the coastline of the Atlantic Ocean. And what they did was they started to test how good their long-term circulation models of the ocean were based on where people reported finding these small rubber duckies showing up in the ocean. So these things were floating in the back of my mind through my PhD. And of course, I never thought about them during my PhD. They came up over the period of a few years later. So what I want to talk about today is how I kind of piece those together with some research that we're doing that also involves photochemistry of suspended sediment. And I'll talk about that in a minute. So the basic starting point is if you think back to when you took Intro to Fluid Mechanics in grade school, and as you're thinking about how things move around. So I'll talk about fluids in a second, but if we just think about traffic, there's 2 ways that we can think about cars and traffic. One of them is that we stand at an intersection and we basically think of the cars going by. And so everything's kind of this flux moving by. The other one is that we can also think about movement of traffic by actually being in a vehicle itself and basically being the thing that's moving around within the vehicle. So from your grade school fluid mechanics, you know that we have these different ways. That's usually in chapter 2, right before you kind of fall asleep. That's the reference frames, and we usually skim through this very quickly. So one of them is that we go out to a river with our velocity meter, and we watch the water kind of passing by, and we measure the velocity as it goes by, and that's the Eulerian perspective. And so this is a flux-based or movement-based approach where you're going to stand still, and the movement of the thing going by you is what you're going to measure. You're going to measure basically all of these things going by. The other one is that we have a start and a finish, and we float in orange or something else, and we measure the velocity by this individual particle going through it. We call that the Lagrangian perspective. So we have these 2 ways. Both of them are correct. Both of them are accurate. They represent different things. They're 2 very initial starting points of the way we're kind of framing the problem. So a lot of words. I apologize for one, but I want to get all of our grammar vocabulary in set. So we're going to conceptualize movement based on really predetermining or presupposing how we're going to conceive of movement. And there's 3 ways of doing this, really 2, and then one more that I'll throw in there. So the first one, the Eulerian. So we're going to measure the flux of objects through a spatially bounded area. So we're going to start with an area, and we're going to measure these things as they go through there. And this is the best way to measure things for nutrients or for anything that's very, very small, because we can't really follow that really small thing. So we have to do this based on necessity. And because it's a whole bunch of small things, we're going to use that very commonly for objects or things that lack individuality. So basically, by sampling a whole bunch of these small things, we're basically getting kind of a statistical representation of them. We're going to increase our understanding of the way this system works by increasing the density of observations. So we're going to increase the number of sampling points that we have in that. And what the limitation, the loss of individualism. So by basically throwing everything in and saying water parcels or suspended sediment particles all move this way, we increase our understanding by basically considering these things in aggregate. And it's a natural choice for any type of science that's place-based. So if we want to understand this watershed, then we're going to measure a whole bunch of flux through that watershed. So the other choice is Lagrangian, and that's tracking specific individual identifiable objects through time. So we need to be able to tag something and follow that through space and time so that we can find it again. So again, if these objects are very small or indistinguishable, we can't really use the Lagrangian approach, and so we have to use the Eulerian approach and vice versa. And this is a better way, this is the natural approach that we might use for understanding when individual things have more individuality. So if there's more, if things can control their own motion, then we're going to need to tag these individual things. We're going to increase, in this case, our understanding by increasing the number of objects that we track and track them through time, or we might track them over longer periods of time and space and follow them farther. Synoptic, the way to conceptualize this one is basically satellites. So we're basically going to take a single snapshot in space and time, and that's going to be our source of information, is that one grab. Okay, so one of the important things of this is that really entire subdisciplines within different branches of the natural sciences are predisposed to different reference frames. So ecosystem ecology or biogeochemistry, basically we bracket a watershed and we measure the flux of nitrogen or phosphorus through that watershed. That's by definition an Eulerian approach to understanding the system. The same thing if a lot of behavioral ecology, mapping an individual elephant or mapping an albatross flying over the ocean, that's a natural Lagrangian-based approach. We're going to follow an individual and we're going to see what happens to its physiology through time. So we're predisposed in disciplines of going one versus the other. Now that there's some disciplines that don't have a natural predisposition where this starts to matter quite a bit. So I'm going to take a pause here. Everybody good on this? Remember this from early grade school? Something possibly later. All right, so when we start to look at some classic figures in geomorphology, the most important discipline, we start to see that some of these subdisciplines have natural predispositions towards these different reference frames. So this is Stan Trimble's study of Coon Creek watershed in Wisconsin, and he's studying the sediment flux through this small watershed. And basically the sediment sources are inputs, the sediment sinks are outputs. This is basically just a budget, and it's a naturally Eulerian. We're going to bracket the space. We're going to measure the fluxes going in and out. This is fish migration through the Columbia River. So this is upstream bound salmon, same thing. So basically what are the stocks and fluxes of those salmon? And then biogeochemistry, this is actually the inputs and outputs of carbon in the Ocoee watershed in Georgia. So same thing, we're going to bound an area, we're going to just study the overall fluxes of these things. So the whole approach is box the system, quantify the fluxes. And one of the important things to note on this is your representation or your mental model is based on where you draw your edges to measure the fluxes in and out of. So one of the things to notice with like the fish migration, Lower Columbia comes in and they're measuring the actual fluxes at very specific dams. And so one of the things that they don't necessarily get is, well, was there much loss of salmon at tributary junctions, where it turns out that there's an enormous amount of bird predation in the Columbia system that we've been missing because we've been measuring our losses at particular places that lose some of these potential other sources of predation. So when we presuppose these boundaries of the system and measure the fluxes in and out of that, we're almost predetermining what our answers are actually going to be. So one of the ways to get around that is by following an individual through it and seeing what it turns out. So we're going to do that a little bit more here in a second. Okay, so the other way is to look at Lagrangian approaches. So two of the same examples. Lagrangian sediment transport. So this is basically putting a tagger onto an individual sediment particle, putting it in the stream and finding it somewhere downstream, which the group in this room knows very well. And so some of the classic ways that we represent this are time since erosion. So we're going to measure time on the x-axis, the transport distance on the y-axis, and we're going to start to develop certain statistics such as the rest periods, the step lengths, and we're going to come up with a way of describing sediment movement using these different description kinds of characteristics. We do the same thing with fish, where we actually tag fish and watch them move around. And so in this case, what we see is fish number 1 moved a certain distance right away and then never moved again. Fish number 3 kept moving, then stayed still. And fish 2 kind of did a little bit of everything. But one of the things, if I toggle back and forth between these, is the way that we're presenting data is completely different between, they're the exact same problems, but the way we actually just even present the data changes based on which reference frame we actually have. So we end up studying the same thing, but talking about it in completely different ways and analyzing data in completely different ways. Now, this is the one great example that I have. So this is by a series of geomorphologists studying the Amazon River. Bob Mead, this is from one of his quotes that's in a paper related to this. This one was by Tom Dunn. And so he notes in the discussion of this paper, they're looking at sediment fluxes in the Amazon River, and they're looking at the stores and fluxes. And one of the things that Bob Mead noticed was when they started to study the sediment fluxes was once sediment got deposited into the floodplain or into a gravel bar, statistically, that sediment particle would sit there for decades, centuries, or even millennia before it got remobilized. But the way that they were actually studying it, they were studying basically the sediment flux within the water system itself. So they basically, by drawing a boundary and looking at sediment fluxes, once that sediment was out of its picture, we didn't care about it anymore. And it was gone. It was exited from the fluvial system. And what Bob says is basically, that's kind of a weird way to think about it. So anyway, I'll read this, and I apologize for reading it, but it drives home the point. Would geomorphologists have focused on individual sediment particles' movements beyond the reach scale, emphasis would have inevitably focused on sediment storage and thus the processes that sediment undergoes during storage. Instead, the Eulerian or reach-centric approach dominated, and so did fluvial geomorphologists' fascination with fluid mechanics. So if you're only looking in the river and you're looking at flux, you basically want to understand what causes flux. If you follow an individual sediment particle, you inevitably end up on a floodplain, and you end up on that floodplain for millennia. And so if you're interested in sediment, you want to know, well, what's happening while I'm sitting there on that floodplain the whole time, because that's where the duration of time is. So basically, we've ended up studying, people like myself have ended up studying fluid mechanics and calculus and those types of things instead of more of a classic kind of weathering approach because of this different reference frame kind of an approach. So this was my grandfather, Stuart Fisher, wrote this fantastic paper that's never cited called Creativity, Idea Generation, the Functional Morphology of Streams. It's a brilliant little paper, especially for PhD students to read, of just how do we come up with ideas? And it's just this wonderful kind of experiment on how do we come up with ideas. And the other book that was always required reading for me was Thomas Kuhn's The Structure of Scientific Revolutions, the same thing of how do we come up with ideas and how these new ideas start to pivot entire disciplines as well. So for the PhDs that are out there in the ether, go thou and read, I guess is the thing. So, all right, so I'm going to put that into a side bucket for a second. I want to pivot over to what kind of drew this out was, so I study sediment movement in streams and rivers, and a group of chemists came and started to ask us questions because they were very interested in how pharmaceuticals were responding in the environment. So we're getting an increasing number of pharmaceuticals, herbicides, fungicides, things like that. And the vast majority of them photodegrade. So if you take a pharmaceutically active compound, you put it within especially ultraviolet radiation, that parent molecule degrades very quickly. And so the longer that basically if we fertilize a lot of crops or if we end up with disposal of pharmaceutically active compounds in the environment, one of the questions is how long do those sit in the environment before they degrade into inert molecules? So one of their questions was, we know that we have a very large amount of these compounds in streams and rivers. And one of the questions was, do they degrade before they reach the estuaries, is one of the questions. So they said, if we basically put these into the water, how long will they sit in ultraviolet radiation before they get to an estuary? And is that enough time for them to degrade? I go, okay, so I sit down with my math And I calculated out and I say, yes, they will degrade. It's fine. And so create more pharmaceuticals, I guess, was my answer. So that was the starting point. Now, the way that we normally do this is to come up with that decay curve of how quickly do the pharmaceuticals degrade in the environment. We take them to photosimulators. We basically, with each one of the chambers, we put different amounts of suspended sediment in it. We shake it up. We put different wavelengths on the light. Then we put it into the photosimulator. We let it sit for different amounts of time and see how much is left. So it's a very simple set of experiments that we run. And this is the way we've always done things. You basically put it in, you take out each one of the samples over a period of 1 hour, 2 hours, 3 hours. You see what your decay rate is. Bob's your uncle, you're all set. And then we started to think a little bit more. And what we started to think about was the fact that these these compounds are not just sitting in water consistently being exposed to light, but even something like in a meander bin, if you imagine that you're a pharmaceutically active compound, if you're a molecule, even in one meander bin, you're actually being advected into this helical flow. So you're actually being advected into the photic zone and out of the photic zone, and you're constantly being cycled in and out of that. Same thing if you're any kind of a compound that's attached to a suspended sediment particle, you're also being advected through photic and aphotic conditions constantly as you're going around one meander bin. So what we know is that compounds are not exposed to light consistently. We know that basically the light switch would go on and off constantly, but we don't know if that matters. And most of the pharmaceuticals or the people researching them said it doesn't matter. It's just cumulative time. It's basically just time. And then this paper, I'm not a chemist. This looks like chemistry though. This paper came out in Nature a few years ago where a group of hydrologists actually, they documented what they call a vampire molecule. And usually the photochemical reactions are one directional. So you take a parent molecule, the pharmaceutical, you expose it to sunlight or UV radiation, and there's a one directional pathway where it degrades over time. And what they documented was that some of these compounds, when they were exposed to sunlight, were being degraded. But then when they were taken basically out of sunlight during nighttime, or especially if they were advected into the hyperrheic zone out of light, some of them were reverting back to the parent molecule. And so what that means is it's not just the turning on and off the light actually matters, because you may degrade some and then you go back to the parent molecule. More problematic since this paper came out and as people started to pay attention was if you degrade in the presence of dissolved organic matter versus the lack of dissolved organic matter, basically the children molecules turn into very different things. And so if you're in the presence of light and dissolved organic matter, you turn into one set of subcompounds. If you are basically in the dark, you stay as the parent molecule. If you then are exposed to light, then back to dark without DOM in the environment, you revert back to the parent molecule. So the pathways, the chemical pathways, are very dependent on the sequence and timing and location of light and dark. So we had all of this in our mind. And as all of us are doing, we're always looking for funding. So we're trying to figure out a way of how do we marry these two concepts into things. So there was a group of hydrologists and estuarine scientists at UC Berkeley, and they had developed a sensor pattern or a sensor package that was basically, it's like a small buoy. And so they would throw them out into the estuary, and they would, the way they would track them is they would have their name and phone number on them. And they would say, when you find this, call us. It's like a rubber ducky with a name tag. And they would measure one or two particular constituencies. And again, they were trying to do it with, again, this is a kind of Lagrangian technique, and they're trying to increase the resolution of their data collection by increasing the number of things that they're throwing out into the environment. We didn't like their technique, and we thought we could do it better. At the time, we thought we could do it better. And so one of the key things that we wanted to do, since we were interested in this vertical dimension of things, we needed a sensor that was going to be neutrally buoyant, that was going to be long-term, because we wanted to study long river reaches. So this was a multi-day and multi-parameter. We wanted to measure a bunch of things, not just one or two things. So I got a grad student, Scott, that's up in the top left, and he created the hydrosphere. Dramatic pause. And so the hydrosphere, it's basically a beach ball. We fill it with water. We try to calibrate the temperature of the water that we're putting in. That would be the temperature of the water that's going to be there. And then very cleverly, what he has is a transponder on it. So it's always beeping a certain radio frequency. It's got basically a cell phone signal in it as well. So if it actually gets up to the surface, it can tell us where it is and it can download some of its data. And then it has a series of different modules where we can actually put different sensor types onto it. One of which is light. We get kind of directionality, a DO sensor, and a few other things like that. So Scott worked on it, started a company to build these and sell these. Company went bankrupt, and so Scott's back in academia. So that's the way these things go. So Scott developed them, and then John is another PhD student, and so John actually worked on it for his PhD as well. And we've deployed them over a series of different types of rivers, and I'm going to show you some of that data as we go. So what we wanted to do was look at these three different reference frames of collecting data. So the synoptic is basically a snapshot in time. And so the way that we did that was we worked with a group at the University of Wisconsin that has basically a speedboat that has a bunch of sensors on it. And so in 1 hour, we would drive as fast as we could all across this landscape and then have a representative synoptic sample of light, dissolved oxygen, a bunch of water quality conditions as well. The boat's really clever because it actually is sucking water in, pushing it through a sampler, then pushing the water out. So as you're driving, you're basically getting continuous sampling. And then it has a high resolution GPS sensor as well. Then we did the more traditional way of the Eulerian. We set up a series of basically static buoys, and then we had sensors on those buoys as well. And this is what the U.S. Geological Survey, in our case, has in a bunch of different places. So that's the way, typically, that's basically we sit still, we look at the water as it goes past us. And then we released the hydrospheres through these river reaches as well. We did a whole bunch of different channels. We worked on the upper Mississippi River to get a really big river. The Neuse River is about a fifth order river in eastern North Carolina. We did some smaller ones as well. And then what we would do is release buoys either collectively. We just put 4 of them into the river. We'd release one at a time, once every hour or so. We tried different ways of releasing them. Okay, so what the buoy does is you put it into the river. It's neutrally buoyant, so it disappears. And then you follow it with a transponder and you try to find it. And after a set period of time, it blows a CO2 cartridge, fills itself up with air, and it shoots up, and then it starts pinging at you and telling you where it is. But because there's a lot of sensors, which means that each buoy costs too much, we also stay in the boat the whole time with the transponder. We run around and try to find it, make sure that we can track it down. So we would let these go for a couple days at a time and basically spend the 48, 72 hours on the boat, just making sure that we could actually find them. Just to sympathize with field workers of how painful this is, typically they get caught in large woody debris jams, and they always get caught way underwater. And this is several $1,000s. And so we try to take apart the debris jam to find it. So in this case, Scott has he can hear it. He knows it's there. It's beeping at us. It beeps a certain frequency, so we know which one it is. And so what we did find was we couldn't go underwater to get it because it's dangerous down there. But there's people who pull dead bodies out of places like this, and it turns out they need to practice. So they loved us, and they would actually come out and rescue for their training exercises. They would rescue our balls out of these different places. It was a riot. They would say, "Are you going to go out and do something again?" And they'd come out and they'd find them and set them free. So that's how we figured out how to do this. So I'm going to go through a set of data, a couple of these subsets. So we would, in this case, the fixed place, so the Eulerian sensors, we measured in 2 places over the entire solar day in this case. So this is one of the places, 3 different depths on this over a period of days. And so what you're basically seeing closer to the surface, you're getting a lot of light coming in. As you go deeper, you basically lose light. And so using those 3 different dates, 3 different depths, then we get a light extinction coefficient. So how quickly do we lose it? So the more turbid water is, the faster you're basically losing light. The clearer water is, you basically get a longer curve on that. And we calculate this as the decay curve or the light extinction coefficient. Then we come up with a photic depth. So one of the things to notice is this is a single place in time, a single place over the period of 24 hours is at this single place, the light extinction coefficient is actually changing over the period of 24 hours or over the period of the photic day. So normally what limnologists will do is they'll go out with a Secchi disk, which is like the first thing you learn in aquatic ecology. They'll lower it and they'll do it at a point in time. And that's basically the photic depth is and the extinction coefficient is. And what we found was that there was actually a lot of variability in rivers for the photic depth and for the light characteristics of this water column. And that matters a lot because increasingly rivers are being studied as lakes. They're just lakes that move. But it turns out for a lot of conditions, we're actually changing the photic characteristics of rivers over the period of 24 hours, even when there's not actually a flood or spate happening in any way. So, so if you imagine that you're a sediment particle or you're a compound molecule, you're moving downstream, depending on when you move through an unshaded area, it might be night, so there's no light, or it may be a time in the day when there's a lot of photic depth to it, so a lot of light getting through the water column, or a little bit of light getting through it. So when you pass through those reaches is going to actually matter. So what we then did is we had basically these different Eulerian places, and then we started to just release these hydrospheres out. And then this is when it started to get really interesting, because if you imagine being a neutrally buoyant water parcel moving downstream, which path you end up going on starts to affect how much light you're exposed to. So if you get advected to the outside of a meander bin, and a meander bin is shaded, for that entire meander bin, you're going to basically see very little light. If instead you're kept into the middle of the river and you're towards the water surface, you're going to see a lot of light. So the individual flow paths we thought might actually matter a whole lot. We might actually get, for the exact same reach, we might get very different samplings of that. So the other thing that as we started to get some of the data coming in, what we realized was we would get a flow path and there would be a depth that hydrosphere would actually go through. Since it's neutrally buoyant, it's basically sampling these different depths depending on where it's advected. And that means that individual water parcel would basically see a whole bunch of different light as it's moving downstream. And it may actually be passing in and out of, when it's up high, it's actually going to be exposed to light. When it's down low, it's going to be in the aphotic zone. So basically you're getting these short duration or long duration intensity magnitude types of events or light events. And so we started to think about these light events kind of like hydrologists, so frequency duration curves of what's the frequency duration intensity of a light event that this water particle is actually exposed to. So this is some of the data. On the left is the Mississippi River, and on the right is the Neuse River. This is the river in North Carolina. In both cases, the Mississippi River goes super deep. We actually made this slightly positively buoyant to keep it a little bit higher up so that we could recover it more easily if needed. But even then, it was basically being advected through between 0 and 4 meters of depth. On the Neuse River, it's a shallower river. It has really big sand dunes. And so what we think was happening, the reason that it ended up so frequently in the top 2 meters, is that even when it got kind of pushed down or advected down to the bottom of the river, that then it was basically rolled and kind of launched back into the upper elevations of the river. So we have these two different sampling frequencies. This is the period of time when we lost it on the Mississippi River, and then it showed back up. And so we start to sample a little bit of a different area. Now, this is on the upper Mississippi River, and just to give a sense of the kind of conditions that one of these buoys or one of these hydrospheres would be advected through. So we're basically this was actually a side channel that one of them got advected into. And you can see that one of the tributaries is basically creating this really big suspended sediment plume. The natural river has a lot of DOM, but it tends to be more clear than the tributaries. So if one of the hydrospheres got advected into one of these areas with a lot of where the tributary influence is, then the lights would get turned out, even if it's in the top meter or so. So again, these individual pathways start to matter for the light. All right, so we have each color is a different flow path. This is a single reach of the Mississippi River. Each one of these graphs is a particular flow day, but each color is a different hydrosphere. And one of the things to notice is that only in one case or two cases for the first few hours did all three make it, then we lost one. For a few hours, this one made it, but otherwise we always lost a sphere. So sampling tended to be a big problem. And the first one on June 16th, we lost two out of three. But PAR is photosynthetically active radiation. So this is basically how much light in the photosynthetic wavelength we're actually doing. And so on June 16th, sphere number 3 basically went 7.5 kilometers and only really experienced light twice. So it was basically dark for the sphere for its flow path, and then it got basically a burst of light, and then it basically stopped getting light. On June 23rd, looks more like a hydrograph where there's a whole bunch of different light events that are happening. June 29th, there's long periods. So again, we have, these looked like hydrographs to me. When I pulled these data down, I can start to sample these and think of these like hydrographs. But one of the things to notice is that only in a few cases, so for this one, did the three spheres actually experience very similar light signatures. And other than that, they basically experienced completely different light conditions as they went downstream. So when we pull all of this together, one thing that we did was we calculated decay curves. So basically, how much light is there as a function of depth? But instead of this being sampling a single point in space, this is basically a light decay curve sampled over all flow paths for this one reach of river. So it's basically a Lagrangian sample instead of an Eulerian sample of the light decay coefficient. And one of the things that we found out of this is that the light extinction, again, changed through the course of the day. So we could use the same sampling approach to show what we thought we found in Eulerian sense, that one sample during the day doesn't, even when the flow doesn't change that much, you get very different light conditions over the course of that day. The other thing that we've started to play with is that's turning out to be really hard is we've taken the light signatures, so we'll take July 5th, and we basically have how much light is being exposed and lights turned off, then lights turned on, then lights turned off, going like that. We take this light signature and we then go back and we take different compounds, and instead of just turning on the light simulator and turning it off after 12 hours, we basically have a student sitting there looking at and say, okay, the light's going to be on for 37 minutes. And then we turn off the light, and then we wait 42 minutes, and we turn it back on for 3 minutes, and we turn it back off. So we're trying to recreate, and we're trying to see what the effect of the different light signatures is on the end quantity of compounds that are actually coming out, and whether we could actually measure that difference at the downstream end of some of these reaches. So far, there's no effect. And part of that is because of something I'll get to in a second. So, well, let me go back. The reason that it's turning out to be that is on this top part, when we sample these rivers over and over and over again, they're dark. So from a photochemical sense and even from an ecological sense, the vast majority of time that these hydrospheres experienced the river, it was dark. So most of the time was below the photic zone. And we're still trying to figure out what that means for ecological, so for like fish behavior and things like that. But what we basically, fish are finding areas not just limited by hydraulic habitat or by temperature, but they're also needing to find, if they need light or if they need vision, they're needing to find places that have light actually available to them. And we think that those are much smaller areas of the river than we've previously thought before. Okay, so that's kind of the science part of things. I then gave this presentation to an equally bored group of people a couple of years ago, and one of the people in the group was a lawyer, and they do come to science talks every once in a while. And so afterwards, he started to talk to me about legal implications and things like that, which what I'm going to just briefly talk about now. And so his kind of mental experiment that he ran was, okay, so we could think about this building. If we wanted to talk about the temperature in this building, how would we potentially measure the temperature in this building? And we could do it fixed site. We could basically put thermometers in fixed places in the building, which is what we do. We probably have a thermometer in this room and it kicks on the air conditioner. The other thing that we could do is we could put thermometers on people when they come into the building, and they would basically walk around, and we would get an individual based sampling of the temperature in the building over the course of whatever time period that person. We'd only get a sample for what spaces that person did. But that moving one, or Lagrangian, that would actually be the temperature distribution in the building that's actually experienced by an engineering student or a geography student or whatever else that is. They're both accurate, but we've typically thought that this was a more accurate way to measure the temperature in a building than the moving one. There's also the snapshot, which is we basically get a thermal camera, and every once in a while that thermal camera takes an entire picture of everything, and then we get an entire distribution. All of them are accurate, but each one is for a different purpose and has different implications. Now, this started to get a little bit more serious when COVID happened, because if you think about, there was basically the virus was in the air and on surfaces. We could measure that at fixed sites. We could measure individuals, are they actually getting exposed to that virus? or we could do kind of a snapshot-based approach. So the actual exposure to viruses, how we measured that started to have much different implications than just temperature. What this law professor, Jim Salzman, started to do was he said, well, if we can start to measure the distribution of stressors, and we can start to measure the susceptibility to those stressors, there's actually a legal pivot that happens that has big implications under US law practice. So what he and I did was he said, okay, you've got buoys, these guys, the Berkeley guys have this little cylinder, and we went and talked to sensor people. And we told them, like, if we could measure certain things, they said, just project 10 years. They said, yeah, we have sensors now that we can put into air ducts that will basically measure air quality and be advected through air ducts and sensors. So they're called dust motes that they do, and they'll measure certain things that you want. So they said, okay, just imagine that you're going to have sensors that you can put onto people on their cell phone, whatever else it is, that will constantly measure ambient environmental conditions for something. So you've got that. Imagine that you've got that, and you've got people actually wear Google glasses, or they wear something like that. So each individual now has a sensor that's telling them the environmental conditions around them. So that information is available to the individual. And we also now, we're sampling people's DNA, and we're able to tell them their susceptibility to different types of diseases or their susceptibility to environmental stressors. So basically imagine that all of this information is available. So we're going to have continued improvement in environmental measurements. We're going to have continued improvement in tracing pollutants in the atmosphere and the environment. We're going to have continued improvement in genetic insights of the individuals in terms of their susceptibility. And we're going to have Google measuring everything that we do, where we go and what we're exposed to. So Google will now know what pathways we're taking on a day-to-day, hour-to-hour, minute-to-minute basis. So I'm going to pull off to a legal article about genetics. So this growing data set of genetics indicates that there are significant genetic susceptibilities to ambient air pollutants in the general population. That is, people are differentially susceptible to air pollution. The issue then presented is whether and how this information can and should be used to better protect genetically susceptible individuals. We're reaching the point of scientific understanding where we can start utilizing genetic information to tailor our environmental and public health policies to better protect susceptible individuals. So what Gary Marchant was saying is, if we know genetic susceptibility, we can start to regulate the polluters more effectively, which is what basically everybody was assuming. What Jim Salzman, this legal scholar, pointed out was there's also this legal doctrine in the US called come to nuisance. And that idea is if there's a plant, manufacturing plant, and it's emitting pollutants, or it's making a lot of sound in this case, and you as a landowner buy a house next to this very large factory, you can't sue that factory for noise pollution because you came to the nuisance. You moved to a place that you knew was producing this noise. So you came to the nuisance, so you're not able to sue them. So what Salzman said was, okay, so let's say that all of us know our genetic susceptibility, or at least the Institute of Health, the Surgeon General does, and they're tracking our movement now. So they know how susceptible we are. They know that we're moving around. So they're basically mapping our movement on a day-to-day basis. And they know the spatial and temporal distribution of pollution. Then instead, could you basically flip from regulating the polluter to regulating individuals and saying that, well, you knew that you were susceptible to air pollution, you knew where the air pollution was, and we know from your spatial data from wearing these glasses or using your phone that you were actually going through an area that was polluted instead of choosing a pathway that was away from pollution. So you can't, we are not going to regulate this polluter because 30% of the population can move in a way that gets them around that polluter instead. So instead, it actually may be flipping it, what I call the Lagrangian flip, which he didn't like at all, but I like it a lot. And the idea is that we could actually be regulating individuals more aggressively rather than regulating the polluters more aggressively by having this increased resolution of data. So you can't sue because you knew that you were susceptible, you had options for movement, and the data actually shows that you chose to do that thing anyway. It's like having peanuts on an airplane. You shouldn't tell, if you're allergic to peanuts, you should choose an airline that doesn't serve peanuts. And if you can't get angry at people eating peanuts on an airplane because you knew that you were susceptible. So just to close things out, For research, I think one of the big things that we learned out of this is just at the very start of a research project is whatever assumption you have about the spatial or temporal distribution, again, just shifting it a little bit and just saying why to think about how we're thinking about the system, whatever the presupposed boundaries of the system are, whatever the presupposed way that we're collecting data. And then the other one is that I'll just kind of plug this paper and these books, or this book about just thinking about how these different types of observations are the grist that start to pivot not just disciplines, but also thinking about cognate disciplines of things like environmental law or things like that. And with that, I'm happy to wrap up and take any questions that are out.